Hi, welcome to Chapter 1, Introduction to Physiology. So in this chapter, we're going to start off by defining physiology. We'll then review the different organ systems that make up the body. We'll talk about the difference between function and process, and then introduce the concept of homeostasis, which is something that's going to come up throughout this term. And then we'll end with science of physiology. So what exactly is physiology, this class that you're taking, and how is it different from anatomy, a course that you guys all completed? So in anatomy, you focused a lot on the structure of the body, and there wasn't as much emphasis on function. Physiology, on the other hand, is building on what you learned in anatomy. So please remember what you learned in anatomy. Because what we're going to do is we're going to take that structure that you learned about and we're going to talk about how it works. So we're going to look at function and process. So we're going to ask why and we're going to ask how. Another key word in physiology is the word normal. And that's because in physiology, we're going to focus on the normal function of the body, the normal processes. When things are abnormal, that's pathophysiology, which you will take in the future. So why is it important that you learn physiology and you learn it well? Well, because as nurses, you're going to be learning about disease states, illnesses, altered states, and what normal should be and how you're going to treat that disease state. In order to do that, you have to first understand what normal function and normal body processes look like. If you can't recognize normal, how are you going to recognize abnormal? And how are you going to know what you want your body to then return to? So I hope that makes sense. To really understand physiology, we have to understand the levels of organization of the body. So we have our cell, that's the smallest structure capable of carrying out life processes. Now, if you look at this figure at the bottom, you can see it shows levels of organization from simplest to more complex. For example, we have our atoms that, when we put together, makes up your molecules. And then we put molecules together, it'll eventually make up a cell, and then we have our tissues, organs, organ systems, etc. What I want you to notice here is that the science of physiology encompasses the broadest range. So that makes physiology really interesting, I think, because you're going to look at how our body works as a whole, how different organs are affected by something, and then what's going on even at the cellular and molecular levels. But that also means it's a lot more complex. Now, here are the 10 organ systems we will be discussing in this course. So the first that's listed is the integumentary system, which really is your skin. It forms that protective barrier from the outside world. Then we have your musculoskeletal system, your muscles, your skeleton. They are there for support and movement. The next four systems allow exchange to happen between the internal and external environment. So the respiratory system, you're going to exchange gases, bring in oxygen, expel CO2. Digestive system, you're going to exchange things like nutrients, water, waste. And then we have your urinary, where we're going to exchange water and waste as well. And the reproductive system, where you're going to exchange your gametes, your eggs and your sperm. The next four systems extend throughout the body. So the circulatory system distributes blood through our blood vessels through our body, and the materials within them, like red blood cells that carry oxygen, nutrients like glucose, gases like oxygen, which I already mentioned. Then we have our nervous system and the endocrine system, so neurons and hormones, and these systems really help coordinate your body function. And finally, we have our immune system, and that's really going to protect us from ourselves as well as from foreign invaders. Now, function and process. Function explains the why. We also call this the teleological approach. Process or a mechanism describes the how. It's also known as the mechanistic approach. So if I were to ask, why do red blood cells transport oxygen? And I answer because cells need oxygen and red blood cells bring it to them, that's the why. If I answer oxygen binds to hemoglobin contained in the red blood cells, that explains the how. So again, in physiology, we have to ask both of these questions. Why does something happen? And then how does it happen? We call this, looking at both of these types of questions, an integrative science. So we're really integrating why and how things work. So homeostasis, like I said earlier, is a concept that's important for normal physiology. And it really is the ability of the body to maintain a constant internal environment by returning changed conditions to normal. So I like to think of homeostasis as when your body is happy, when your body is normal. That does not necessarily mean everything is in balance, that everything is equal. It just means our body is the way it's supposed to be when everything is working correctly. Again, it does not mean things cannot change. So parameters can change, but when we are in homeostasis, we have responses that the body will undergo to return those changed parameters back to normal. So I've been saying parameters, well, what am I really talking about? Pretty much everything in the body. So things like osmolarity, temperature, pH, gases like oxygen, carbon dioxide, ions like sodium, potassium, calcium, our hormones, just all of these things have to be maintained at a homeostatic level for our body to function properly. They can change, but then we just have to return it back. If altered parameters are unable to be restored to homeostasis, that's when illness and disease can occur, and so then that would be the study of pathophysiology. So how do we know all of this? Well, somebody asked a question, and they designed an experiment. They collected data, they interpreted the data, and then the experiment was replicated and eventually became theories and models. So I want to talk about three things about designing an experiment. And the first is the independent variable, then the dependent variable, and then your control. So the independent variable in an experiment is what you, the researcher, changes from group to group. The dependent variable is what you, the researcher, measures. And then the control is the group that you really kind of leave alone. So let me give you an example of an experiment, and I will identify these parameters or these variables. So what I'm going to do is I'm going to divide the class into three groups, A, B, and C. In group A, I'm going to ask them to study for 2 hours every day for next week's quiz, your first quiz. In group B, I'm going to ask you guys to study for 2 hours on Saturday. And then group C, the control group, I'm just going to leave you alone and say, hey, there's a quiz next week. Then you'll come next week for your quiz and you will take it and I will grade it. So in that experiment, the independent variable, what I measured, what I changed from group to group was your study time, hours of study time. The dependent variable, what I measured, was your grade. And hopefully what the experiment will indicate to me is that when you study a little bit every day, you're going to do or perform better than if you just studied a little bit one time a week. Now, when we do human experiments, interpretation of the data can be a little bit difficult because of things like genetic variability. So no, you're not going to find two groups of people that are genetically identical that you can do experiments on. So we know that our genes play a big role in a lot of things, and so we have to kind of take that into consideration. Then we have the placebo effect and the nocebo effect. So the placebo effect is a positive psychological effect. Nocebo effect would then be a negative psychological effect. Let me give you another example of these. So let's say I have a pill, a blue pill and a red pill. And I give the blue pill to a group of people and I tell them, I'm doing an experiment or I'm testing a drug for, hypertension. And just so you know, this drug is in its final trials and it's been performing really well. And people have also reported not only, their blood pressure going down, but they've also had a lot more energy and they feel just really good. The next group, I tell them the same thing that we have a hypertension drug we're testing, but I also tell them that, people have been reporting that they've felt very fatigued and just really lethargic and, maybe a little bit nauseous and tired, etc. Now, in reality, I gave both groups the exact same pill, but what I changed was what I told them. And what will happen is people who I told like positive things to, they'll actually report that they felt great. So that's the placebo effect. And the group that I said would be tired and lethargic and not feel that great, they'll actually report that. Not all of them, of course, but a good number of them. And so that's the negative psychological effect. So, as humans, we all have kind of expectations. And so, those expectations and our thoughts can really impact our body's physiology. And then finally, ethics. We can't do whatever we want in humans. Now, if we have these considerations to take in human experiments, how do we control for that? Well, it would be really how we design our experiment. So the first way we can design our experiment is to do a crossover study. A crossover study is where the control group in the first half of the experiment will then switch places with the experimental group. So group A and B, we have group A, we have group B, we do the experiment, and then we're going to repeat the experiment again with the same group of people, but we're just going to switch them. So now group A will be group B, and then group B will be group A. So the group who got the red pill in the first study will now get the blue pill. A blind study is where the subjects are blind. So the subject has no idea what they're getting. So they could be getting the drug or they could be getting a placebo. Now, when I just say a placebo, it means essentially a sugar pill. I'm not really talking about the placebo effect that I talked about earlier. So if you are a participant in a study and you go in and they give you a pill, you have no idea, is this the drug or is this just a sugar pill? So that psychological effect is kind of taken out of the equation. Then we have a double-blind study. This is where the subjects are blind and the researchers are blind. Now, by researchers, I mean the people who are kind of administering the study and doing the data recording. Somebody has to know, otherwise the data would be useless. But the reason double-blind studies are done is so that we take any kind of bias out of the results. So the subject has no idea what group they're in. The person collecting the data doesn't know what group they're in. They just know, one drug is called A, the other drug is called B. They don't know which is the placebo and which is the actual drug. And then the most complex study would be a double-blind crossover study where group A and B, they both don't know what group they're in, whether it's the drug or the placebo. And then once the experiment is done, we switch them and do the experiment again. So most complex. So that's it for chapter 1. It was our shortest chapter, and I look forward to speaking to you guys again. Bye.